we're doing a micro lecture on biomolecules. So biomolecules really, comes down to one thing. They contain carbon. Carbon is in all of these biomolecules. Then this chart right here are the functional groups commonly found on biomolecules. So obviously it's going to make a lot of these molecules polar. And so all of these are polar molecules. Like I said, most molecules floating around in your body are going to be polar. Structurally, cell membranes can have some nonpolar structures. Some hormones are nonpolar, but we deal with a lot of polar molecules. So hydroxyl is going to be an OH. That's very common, as well as phosphatate, carboxylamine, and so on. Classes of organic molecules, and again, organic just means contains carbon. So if you take organic chemistry, it's chemistry that only involves molecules that contain carbon. So we have carbohydrates, lipids, proteins, and nucleic acid. So we're going to go through each one of these. Carbohydrates, you can have monosaccharides, meaning one, just one. I should say just one molecule. Disaccharides, di meaning two molecules, and poly meaning many molecules. So a monosaccharide is going to be one glucose molecule. A disaccharide would be like sucrose, which is a glucose molecule and a fructose molecule. So there are two molecules. That's where disaccharide comes in. And then polysaccharides are going to be carbohydrates like starch or fiber, and there's many molecules involved there. So that's polysaccharides. Lipids, most common, fatty acids, triglycerides are the most common fat used for energy in our body. Phospholipids, it's an example of cell membrane. Steroids, an example of hormones. So lipids can take on various roles in our body. Proteins are basically strings of amino acids. And so that's why amino acids make up the subunits. Polypeptides, you can have a peptide, which are less than 50 amino acids in the chain. And you can have proteins, which is more than 50 amino acids in the chain. And so those are proteins. Last is nucleic acids. Most of you guys have heard of your DNA. It's like the big recipe book for making proteins, and that DNA, I know the garbage truck's going by and my door's open. Sorry about that, guys. DNA is found in the nucleus of the cells. And DNA can give recipes to create messenger RNA and basically all proteins. ATP is adenosine triphosphate. There's also ADP, and we're going to talk about this in length in chapter 3. Adenosine triphosphate, that's the molecule that has potential energy that our cells can harness to do work like move muscles or actively transport things. So quickly, carbohydrates, again, it's basically carbon, hydrogen, and oxygen. So those are the three atoms that are going to be combined in certain amounts to form carbohydrates. Monosaccharide, again, this is showing you a glucose molecule, and it's one molecule. So monosaccharides are glucose, fructose, and maltose. Those are the only three types of monosaccharides. Disaccharides, you're going to have two molecules. And this is showing you sucrose, which is table sugar, regular, what we call sugar. And by the way, this is whether it's honey, brown sugar, white sugar, it's all sucrose. You can also, though, have lactose, which is milk sugar. And I got that wrong. Maltose, which is malt sugar. It's galactose. Okay, so I clearly need to go back to school. And then polysaccharides, the most common starch. This is what we eat, right? This is what we call complex carbs. There's also something called cellulose. And really, we call this fiber. This is a polysaccharide we humans do not have enzymes to break down. So enzymes to separate. Sorry about that. I'm really going off the rails there. Okay, so monosaccharides, disaccharides, that's what we call simple sugars, right? And you'll see if sucrose is added, you'll see added sugars, right? In a food, it'll say added sugars, right? We added sucrose, which is right here, which is table sugar, which is what we call sugar. It's really one glucose molecule and one fructose molecule. It is a dehydration reaction, meaning you're pulling out a hydroxyl on the glucose and a hydrogen on the fructose, and you're gonna pull out water in order to form a bond between those two molecules. So that is a dehydration or condensation reaction. Glycogen would be if we connected a whole bunch of glucose molecules. Thus, it's polysaccharide. It's more than two. And so there's multiple glucose subunits bonded together, and that's what makes it a complex carbohydrate. So again, monosaccharides, note the hydroxyl groups make carbohydrates polar molecules, which means they cannot go through a plasma membrane or cell membrane. And it's glucose, fructose, and galactose are the three types of monosaccharides. Disaccharides, you have sucrose, lactose, and maltose, which are two glucose molecules. There. And then if you want to synthesize into a polysaccharide, you're going to do a whole bunch of condensation reactions, joining all these glucoses together, right? You're pulling out water. If you want to take this polysaccharide, say it was stored as glycogen in the body. This is how we store carbohydrates in our muscles and in our liver. And we want to use this stored glucose. We are going to use water to then separate those glucose molecules. So this is called glycolysis. We'll talk about that in cellular respiration. All right, so how does the body use sugar? Well, glycogen exists in the body as our stored polysaccharides. And again, it's stored in the liver and skeletal muscles. If we want to liberate or use that glucose, we're going to do a hydraulic reaction. We're going to separate those glucose molecules so they can then go into the blood or go into the cells and be used for cellular energy. And that energy transfer is what we're going to talk about when we talk about cellular respiration. How we take the energy out of the bonds of a glucose molecule and basically put that energy into ATP, so our cells can then use ATP for cellular energy. All right, lipids. Lipids are non-polar covalent bonded molecules. That means they're hydrophobic. They fear water. They don't like water. The 5 different classes, triglycerides, this is the most common that we store and use as cellular energy. But there are also ketones, phospholipid. We're making up plasma membranes. We'll just briefly talk about eicosanoids, and then steroids type of hormone that is nonpolar. Lipids are important for physiology. So it's the most prevalent source of energy, the major components of our cell membrane, that lipid bilayer. And we also use it in hormones for signaling molecules. Fatty acids are basically carbons and hydrogen with a couple of oxygen in there. If there are no double bonds, it is an unsaturated fat. If every single carbon is bonded all the way around, we say it's saturated. So this is an example of a saturated. Polyunsaturated, you're going to see some double bonds, so we're missing hydrogens here. And so it's not completely saturated with hydrogens. Triglycerides, there's a glycerol with 3 fatty acid tails. And because of the tremendous amount of carbons and bonds between the carbons, there is a lot of potential energy in these bonds. And there's a lot of bonds. And so that's why fat equals high calories, because calories is really a measurement of potential energy. Measurement of potential energy. Okay, so triglycerides, you're going to have the glycerol and the 3 fatty acid tails. They are nonpolar and they are hydrophobic. Phospholipids, again, this is cell membrane. And as a review, they are amphipathic. Phospholipids are because you're going to have the phosphate polar head and then the lipid tails. So this just goes through the, polar phosphate head and then the lipid tails. Steroids derived from cholesterol. These are nonpolar lipid molecules. And again, we use it in cell membranes. They could be signaling molecules like hormones. And here's an example. We have cortisol, we have estrogen, testosterone. These are hormones as well. And the last, well, second to last, but proteins, which is about 50% of the organic material in our body, and it plays critical roles in almost every physiological process. And the shapes of proteins really matter. What proteins are basically strings of amino acids chained or bonded together. Now, there are a number of types of amino acids, and which amino acid that's bonded to the next amino acid is really going to determine the shape. And we get into that when we talk about protein conformation or shape of the protein. But proteins are strings or repeated units of amino acids. And so amino acids are the building blocks of proteins. 20 different kinds of amino acids. And what differentiates these 20 different kinds of amino acids is really the R group, because every amino acid is going to have an amine group. It's a nitrogen and 2 hydrogens. So they all have an amine group. They all have a hydrogen, and they all have a carboxyl group, right? Which is a carbon double bonded to an oxygen and a hydroxyl group. So they all are going to have the carboxyl. What is different on every single amino acid on all different amino acids is the R group. So let's highlight this R group, shall we? This R group right here is going to be different on all amino acids. And so this is showing you for alanine here, this is alanine, and this right here is going to be the R group, right? If we look at, and that's a methyl group or methane. If we look at tyrosine, it's this R group here, glutamate, this R group here, lysine. As you can see, these are very different R groups. So these are the R groups. They're also known as the functional group. And they are going to make each one of those amino acids behave differently when bonded next to one another. So peptide bonds are what combine two amino acids together. And again, a peptide bond, it is going to be a condensation reaction, right? Because you're pulling out a hydroxyl group on one amino acid and a hydrogen on another amino acid. You're drawing out the water and you're forming a peptide bond between the carbon and the nitrogen. Peptides are generally 2 to 50 amino acids. If it gets over 50 amino acids, we call it a protein. So amino acids, subunits or monomers of proteins. Proteins in all living organisms are going to have the same 20 amino acids. They're just assembled differently next to one another. They're different combinations. And so those different combinations are going to cause the protein to fold or become a different shape. Why? Because some are nonpolar, some are polar, some are ionized. And again, two ionized amino acids, if they're both cations, they're going to kind of move away from each other. If they're a cation and an anion, those two amino acids are going to move towards each other. So these are determining the way that the protein folds or its shape. Last thing is humans can synthesize some amino acids, but others called essential amino acids can only be obtained through diet or food, right? And so this is where vegetarians need to combine two types of plant proteins because no plant protein has all of the essential amino acids. Only animal proteins contain all essential amino acids. So beans and rice, right? Beans are going to have some of the essential amino acids. Rice has the other essential amino acids. So when you eat them together, you get all of the essential amino acids. So all animal proteins have all the essential amino acids. Plant proteins, some have some of them, some have others. The last one is nucleic acid, and nucleic acids are store DNA, RNA for expression and transmission of genetic information. So this would be DNA and RNA here, this expression. But storage, we're talking about ADP, ATP. This stores potential energy. And not just any kind of energy, cellular energy for later use by cell. Our cells can't use glucose. They can't use triglycerides. There's potential energy in glucose and triglycerides, but our cells actually have to transfer that potential energy from glucose and transfer the energy to a high energy phosphate bond and adenosine triphosphate. That process is called cellular respiration. So that's a preview for chapter 3. So nucleic acids, DNA. This stores basically the recipes for all proteins in the body. So DNA really holds genes, and genes are a recipe for one protein. And so if you look at the total genome, it's all of the different recipes for all the different proteins in the body, and that's stored in your DNA in the nucleus of the cell. It's a nucleic acid. RNA is also a nucleic acid. You have messenger RNA, you have ribosomal RNA, you have transfer RNA, and we'll talk about that in transcription and translation. And translation of proteins. Those are nucleic acids, and they are created off the DNA strand. And then the energy transferring nucleotides. So adenosine diphosphate or adenosine triphosphate means it has 3 phosphate groups as a nucleic acid, as well as nicotinamide, adenine, and flavin adenine dinucleotide. I just prefer to say NAD and FAD. NAD and FAD do reduction, oxidation reactions. And we're going to talk about this again, chapter 3, cellular respiration. So important to have NAD and FAD to basically move electrons and protons. That's really what oxidation and reduction is. So again, when we're looking at, say this right here, what type of reaction is it's a phosphate, right? So you're adding a phosphate, so you're phosphorylizing. I probably spelled that wrong. Anywho, ATP, again, serves as molecular energy source for all cells. It stores potential energy, and this is cellular respiration. What do we use it for? Everything. Everything the cell needs to do energy. The last nucleic acid is cyclic nucleotides, cyclical AMP, cyclical GMP. These are messenger molecules. We'll briefly touch on this in chapter 5 when we talk about chemical messaging.